I'd like to thank the Academy and the Crawford Committee for inviting me to speak today, and it's a great pleasure to take part in this exciting symposium. So I'm going to talk about what you've heard about in the other talks, surface immunoglobulin, which is on the surface of the B cell. And just to remind you that this surface immunoglobulin is held on the microvilli and on the membrane ridges pointing out into the exterior. So it's a very important feature of B cells. The structure of immunoglobulin on the surface has been well worked out more recently by cryo-electron microscopy. And just to remind you, and because it's important in what I'm going to say, the variable region has the complementarity determining regions, the CDRs, pointing right out into the environment. But the constant regions are shown here, interacting with Ig alpha and beta to transmit membrane signals to the interior of the cell. And one important thing that we need to remember is that expression is required for normal B cells, which die if the B cell receptor is ablated, and for most B cell tumors, particularly the indolent ones. Sometimes the very aggressive ones that get away from any control can lose their immunoglobulins, but in general, they're present. So again, just to remind you about B cell differentiation and the recombination events that occur to create the variable region, the DJ, the VDJ, etc., isotype switching, which go on in B cell development in the bone marrow. And to remind you also that this is a very dangerous process. Moving around DNA is a dangerous procedure. Mutations, translocations will all disorientate the cell, and these combinations can give rise to tumors. And the advantage of looking at B cell tumors is that you can track them back to the point of differentiation of the B cell. So learn a lot about normal B cells from studying tumors. So the acute leukemias come off early. Chronic lymphocytic leukemia comes off at 2 points, as we showed some time ago, a pre-germinal center point and a post-germinal center point, splitting that disease into two and remarkably showing a very different progression rate in the two. I won't go into that today. That's all published. Somatic hypermutation occurs at this point after antigen engagement, and the rate of mutation is 1 million times background mutations. So it gives you an idea of the disadvantages, perhaps, to the cell of this procedure, but the advantages in producing antibody responses. Follicular lymphoma, which I'm going to talk about, comes off here after the somatic hypermutation mechanism is activated. And after isotype switch, then the plasma cell tumors like multiple myeloma develop. So what I'd like to talk about here is B cell tolerance and B cell responses. And what hasn't been looked at in any detail is the fact that the B cell responses fall into two categories. The first one is the extrafollicular, the so-called innate B cell response. This has been rather left to one side by immunology, including ourselves, for quite a long time. But it forms these natural polyreactive, mainly IgM antibodies. And what happens when a pathogen comes into the organism, there is a need for an immediate response. And this immediate response, this innate response, comes from B cells which are pre-existing, whose V genes are preset by evolution for the kind of pathogens that they're going to encounter. And this mops up and attacks pathogens before the adaptive immune response comes up. And that's the germinal center B cell response that we all know and love, which produces high affinity antibodies. But this is going on all the time. And one of the things about this natural antibody response, as I say, it's mostly IgM, is that it also, many of the antibodies also interact with autoantigens. They're polyreactive, low affinity, aim to scoop up any pathogen in the system, but they also react with autoantigens. This is a price, again, we pay for multiplicity of function. When an organism comes in, there's often a lot of cell death, a lot of apoptosis. There has to be a way of clearing away the cell debris, and these natural antibodies have the second function of removing autoantigens, which otherwise cause too much inflammation. So it connects with the fact that B cells can do many other things. They can produce B cell tumors, they can produce allergy, which I'm not going to talk about, and they produce autoimmunity. So going back to this side of the equation, the extrafollicular innate immune response, what you can get here is if you have an infection like EBV and many other infections, as I said, you get this first response, high levels of these IgM antibodies. And in this case, one of the antibodies that turns on is what Chris has been talking about, the 434 immunoglobulin. It's not the only one, but it comes up in quite high levels. And as Chris has explained, it has anti-red cell activity, which is focused on the N-acetyllactosamine. So what you can get in an EBV infection in an adult, you can get this disease called infectious mononucleosis, a very nasty disease where you get a massively sore throat and many symptoms, and you can get the production of these cold agglutinins. So these cold agglutinins can actually give rise to agglutination. And this is a girl who had a miniskirt, it was cold, and she had infectious mononucleosis, and she showed symptoms of red cell agglutination and complement fixation in the leg. So this is an example of an infection causing an autoimmunity, but that also resolves. When it doesn't resolve and it becomes a tumor, you can get what, again, Chris has described in some very nice detail, cold agglutinin disease. This is when the 434 now is monoclonal. It's a tumor. One cell has become the tumor cell. It's producing cold agglutinins, which agglutinate the red cells in the microscope, but also in the peripheral circulation, where if you've got cold legs, you can get agglutination and complement fixation. This can cause gangrene, can be very dangerous. So you have to keep the patient warm. I mentioned chronic lymphocytic leukemia. This is another disease that comes from this, or one subset of it comes from this side of the equation. The chronic lymphocytic leukemia cells have unmutated immunoglobulin. They come from the 169 gene, which we just heard about, another common gene in these natural polyreactive antibodies. And when chronic lymphocytic leukemia develops, these immunoglobulins are all directed towards autoantigens. And one of the commonest one is the oxidized lipoproteins. And these can feed the tumor cells and keep them going. It's necessary for the tumor cell to have these to keep the tumor going. So this is an example of an autoreactivity feeding a tumor cell. And then what happens is the cell goes into anergy. We've heard a lot from Chris about anergy. The cell can circulate in an anergic state, but is kept alive because it's upregulated BCL2. So going to the other side of the B cell responses, to the germinal center response, this will produce tumors as well. And I mentioned follicular lymphoma. It's a low-grade germinal center-derived B-cell tumor. The patient has a swelling, often in the cervical lymph node. And when you look in the immunohistochemistry, you can see that the follicles are quite well maintained, not very dissimilar from autoimmunity, where, again, you can see these developments of active follicles in patients with rheumatoid arthritis. So what is the B cell doing in follicular lymphoma? How is it kept going? We do know that expression is retained. You don't get follicular lymphoma cases without the B cell receptor. We know that it's undergone somatic hypermutation and that that is ongoing in these tumors. So the B cell receptor is still responding to something. What is it? Is it an autoantigen or is it a mimic? What's driving follicular lymphoma? You can see that these cells upregulate BCL2. This is the upregulation of BCL2 in the germinal center, which doesn't come up in a reactive lymph node, so it's very specific for follicular lymphoma and other B-cell tumors. And of course, the cells do eventually acquire other mutations, particularly in epigenetic modifiers. So there are things that happen afterwards, but what's keeping the B-cell receptor going? It's stuck. There we go. One thing that we've looked at in the surface immunoglobulin is that there are introduced sites for the addition of glycans, and René has just nicely described these sites: asparagine, X, serine, and threonine. If you look at an immunoglobulin molecule, it has in its backbone, due to germline sequences, sites where carbohydrate will be added. And these are important in the half-life of the immunoglobulin and complement fixation, etc. They have a function. But there are generally no sites in the variable region. And we just heard from René that they can be in pathological conditions. In follicular lymphoma, these sites are very specific. They're introduced into the variable region, shown in green here. They're rare in normal B cells or in unrelated B cell tumors. And we mapped these sites to the positions in the variable region, and you can see in blue, this is the heavy chain, the sites tend to be, almost always are, in the CDRs, pointing right out into the periphery. So this is very different from the autoantibodies. These are right in the antigen binding site. And you can see from these sequences here outlined in yellow, the sites can be introduced in CDR1, 2, sometimes in the framework region, but they're different there. But even if you've got one in the framework region, there's always one in the CDR. And they're retained during tumor progression. They're essential for follicular lymphoma. Now, what is that going to do? The sugars that are added to the variable region are also very peculiar. Again, René has mentioned the biosynthetic process that adds sugar to these sites which are introduced by somatic hypermutation. First, you get mannose and glucose in the endoplasmic reticulum. Then the glucose is trimmed to high mannose. And this is where the mannosidase enzyme comes in, takes off those mannoses, leaves a few, and then the glycosyl transferases add different sugars as it proceeds through the Golgi. What happens in follicular lymphoma is that the FC region has complex glycan, as you would expect, but the variable region doesn't. It terminates at mannose. And this is very unusual for a cell surface protein. You don't get mannose-related cell surface proteins, and these mannoses are sticking out into the periphery. I just show this here, that the mannoses are out into the periphery. They're fully glycosylated in the FC region, but not in the variable region. And this was shown by susceptibility to an enzyme called ENDO-H. The surface immunoglobulin, which we isolated, changes its size with ENDO-H only in follicular lymphoma and not in normal B cells. So this is just proving the point. And if you expand that and look at it in detail, and we've done this by mass spec analysis now, you can see if you look at two tumor cells, two patients, FL29, FL14, and track the sites which are mannose-related or complex or unoccupied, which you can do easily by mass spec, then those in the CDRs are mannose-related. In this individual at position N55, and in the light chain N107, both mannose-related. The one in the framework region is fully glycosylated. Similarly, in this case, the one in the CDRs is mannose-related. This one is not occupied at all. So this can all be shown by mass spec. So the upshot is that those in the CDRs are mannose-related. So why is this the case? And what we've been able to show by crystal analysis is that there is a blockade to the mannosidase function. So this is one of the patients, FL14, and here's the glycan shown in the binding site here. And here are the CDRs. You can see CDR3 is right up against this glycan, and so is CDR1. In this case, CDR2 is not so involved. And they're shading this asparagine at 38 and preventing the large mannosidase enzyme from getting in there and processing the mannoses further. So what's binding to these sites? What do the mannoses on the surface bind to? And there are a large number of lectins, which are all well documented, some in the soluble phase, some on the cell surface. For various reasons I haven't got time to go into, we chose to look at DC sign. And DC sign, let's see. There we are. DC sign is the candidate receptor for these mannoses on the cell surface of the follicular lymphoma cell. It's a C-type lectin. It's specific for mannose-related structures, although it can also bind to fucose. It's expressed by these cells, dendritic cells, follicular dendritic cells, and M2 macrophages, which are known to be polarized in follicular lymphoma to the M2 by the presence of IL-4. This is the molecule itself. It has a carbohydrate recognition domain at the end and a neck domain formed of 4 chains which tetramerize. So what happens in follicular lymphoma is a very clever hijacking of this molecule, whose normal function is to bind pathogens, and it certainly binds to HIV. It's hijacked by follicular lymphoma cells. These mannoses can now bind to this molecule DC sign. So this is just to prove the point. These are just cells from cases of follicular lymphoma binding to DC sign, 3 different cases binding to DC sign. Up here, no binding to normal, naive, memory, or germinal center B cells, so this is very specific. And when they bind, this is a cell line actually expressing the mannoses, and these are monocytes, or a cell line expressing DC sign. They increase adhesion, just like anti-IgM. They can form clusters, and this is 2 cell lines which are forming clusters, just to illustrate the point, which are broken if there is an anti-DC sign antibody that goes for the carbohydrate recognition domain added. So this is entirely dependent on DC sign interaction. And this antibody was given to us by Carl Figdor's lab. So what we think is happening is shown here. The oligomannoses up on the variable region are interacting with DC sign on M2 macrophages and on follicular dendritic cells. So how to prove this point? We looked by immunofluorescence. This is multi-immunofluorescence done by our colleagues in Rochester, New York. And here on the left, you can see a section of a follicular lymphoma case with follicular dendritic cells outlined in blue, expressing CD23, and macrophages in red, expressing 163. So this is how it looks in a tumor, follicular lymphoma cells. If you overlay that with anti-DC sign, you can see that DC sign is expressed on the follicular dendritic cells and also on the interfollicular macrophages. So both these are candidates for supporting the follicular lymphoma cells. In fact, the follicular dendritic cell is the more likely candidate because of this result that we had from our colleagues as well, in which they showed that Ki67, these are low proliferation rate tumors, they're low-grade tumors, but you can see proliferation by using Ki67, and you can map that onto the phenotype of the tumor cells. And when they did that, this is the CD23 is in blue again, when they did that and plotted out the distance between the Ki67 tumor cells and the follicular dendritic cells, if they were close, that's less than 10 microns, then there was more Ki67. And this is just 3 cases to illustrate that point. So what we think might be happening is there is some influence on proliferation from that interaction between DC sign and the follicular dendritic cell. We also know that DC sign can mediate a modest signal via surface immunoglobulin in cell lines and primary tumors. This is just showing SIC on the left-hand side. The signal is much less than with anti-Ig. Anti-Ig is a strong signal. DC sign is quite a weak signal. And that might be important because B cells do not like a strong signal. They will undergo apoptosis, even in the presence of BCL2. They also mediate a weak but significant phosphorylation of AKT and ERK. An RNA-seq analysis indicated a similar but lower response to DC sign as compared to anti-IgMs. Thank you. The other point is that DC sign does not mediate significant endocytosis. And endocytosis is, of course, a feature of immunoglobulin on normal B cells. If you're exposed to anti-IG, the surface immunoglobulin is cleared, as it should be for antigen presentation. The same is true of follicular lymphoma. But DC sign does not mediate endocytosis. So it uncouples the BCR signaling from endocytosis and allows a continuous interaction. The other point, coming back to Chris's lecture on the HEL system, our colleague Hassan Umar used the HEL system to look at whether the addition of carbohydrate interfered with antigen binding. And what he showed was that if he inserted a glycosylation site at this position in the CDR1 of the anti-HEL antibody sequence, he could ablate the ability to signal through calcium. And he controlled that by inserting a glutamine instead of an asparagine. So it looks as though it's possible that the oligomannoses are actually protecting the tumor cell from exogenous antigen. And that's probably important in keeping it alive. So what we have here is an autoreactivity created for tumor development. Not going. There we go. Where the T1418 that upregulates BCL2 occurs first, somatic hypermutation then comes into the system. Tumor development then takes away the exogenous antigen signal that was the original signal for the tumor, for the growth of the normal B cell counterpart, and leaves the tumor cell looking for an interaction through its B cell receptor, which it needs to survive. And when it finds the right sequence through somatic hypermutation to add on the mannoses, it can then interact with the DC sign, probably on the follicular dendritic cell, or certainly, possibly, on the macrophage. So what this does, it increases adhesion to integrins. It provides a low-level, possibly optimal strength signal to the tumor B cell. It makes it independent of the original external antigen, and it blocks access to any other external agents. The other thing is we can block this interaction. As I showed, Carl Figdor's antibody blocks it when we talk about cluster formation. It can also be used clinically as a very gentle blocking agent to add to current therapies for follicular lymphoma. And we're planning a clinical trial to do just that. This should stop all these other interactions that the tumor cell needs. So just to summarize, there are autodrivers on B cells. B cells generally are interacting with pathogens on the outside, viruses or bacteria. Two kinds of response can occur. Both can give rise to tumors or autoimmunity. The stimulation by a pathogen causes a chronic expansion of specific and bystander B cells. They're not necessarily specific for the pathogen. Inflammation and anti-apoptotic mechanisms. Autoantigen capture can occur, and further mutations, which we heard about, can then lead to autoimmunity. Under a Goldilocks ideal level of proliferative signaling, B cell tumors can be driven by autoantigens, as I've illustrated for CLL. On the other side, a lectin capture, DC sign, can maintain the tumor cell until further mutations then cause B cell tumors, which are driven in part by autolectins. So just some illustrations of tumors that are in these categories: chronic lymphocytic leukemia, mantle cell lymphoma, marginal zone lymphoma, where B cell receptor pathway inhibitors are actually having good clinical effects. And on the other hand, follicular lymphoma, where anti-B cell antibody does have good effects, we're thinking that anti-DC sign will have additional or perhaps more precise effects on the tumor. So the people who've done the work here are listed. Francesca Falcone, more recently with Georgia Chiodin. Max Crispin and Joel Allen are the glycoprotein experts in Southampton. Richard Burak and Ji Lang in Rochester have done the immunofluorescence. Hassan, of course, we've collaborated with over the anti-HEL system. Lou has been a great discussant. And Carl Figdor, very importantly, put us onto this anti-DC sign antibody, which we're now going to take forward into clinical trials. Thank you very much. I think my time is up.